Hi, I am Ashwin. Welcome to my ePoster. Today's topic is on feasibility study about doped nanomaterials as a contrast agent for X-ray excited optical imaging. So if you look at the preclinical imaging, it is a tool for a translational research. If you look at the cancer-based diagnosis, several imaging modalities have been approached till now to understand the hallmarks of cancers. If you look at the hallmarks of cancer, there are several markers that is actually going to help you in understanding how the tumor is getting progressed and how we can diagnose for various imaging modalities. So in order to understand the translational research, it is quite important to transfer any drug in order to convert from clinical to preclinical, preclinical to a clinical imaging. So for this process, it is always important to understand the capability of your drug. So this capability of the drug can be diagnosed or can be predicted or tested using a preclinical imaging system. So it's quite important to understand what is the importance of a preclinical imaging in terms of the drug discovery process. If you look at the preclinical imaging, there are two important aspects that we need to understand. One is how to monitor the disease and the monitoring the progression of the disease. Because as we know, the cancer is going to convert from normal to a metastasis stage. So the progression has to be understood and how this drug is going to be treated for the cancer. And second important point is when the drug is being injected to the patient or any preclinical imaging, and what is the efficacy of the drug. So the efficacy of the drug has to be determined based on the preclinical imaging. So in order to convert any drug from a bedside to the bed, it is very important to understand how the preclinical imaging or what is the aspects of how the drug is going to get response in the human body. So if you look at the preclinical imaging, the bench wherein we develop the nanomaterial and then we go for a clinic sector. But then the preclinical testing wherein it tells you whether this drug is going to give you an efficacy in cancer progression or it's going to treat your cancer for various cancers like colon cancer, breast cancers and everything. But the ultimate problem is that whether the contrast agent and the imaging system is going to fuse together, but what is the problem behind it? As we know, there are several contrast agents that have been working till now. So for example, we can see the FDA approved contrast like iodine that is used specifically for contrast computed tomography, and then fluorodeoxyglucose that is specially for positron emission tomography, and we have optical imaging. But what is the problem behind these contrast agents? If you look at these conventional contrast agents has some problem if we try to understand or if we try to diagnose these tumors in a mouse models or human patients. The problem is if you see this example of a CT image, you can see the tumor progression with a contrast and without a contrast. But if you have a contrast, it is quite clear to understand what is the volume of your tumor and how it can be diagnosed in a CT image. So ultimately we need to have a varieties of imaging modalities has to be fused together to have a hybrid imaging modality so that we can have the structural changes as well as the functional changes of your the tumor area. So if you look at, there are several hybrid imaging technology wherein we can try to fuse an anatomical imaging as well as the molecular imaging. So the anatomical imaging, basically we use different types of systems, imaging systems like the computed tomography, magnetic resonance imaging and the ultrasound imaging. So if you try to combine the anatomical imaging and molecular imaging fused to form a new type of an imaging technology, and that's what we are going to deal today. Another important aspect is it's not only the process or it is not only the development of an imaging modality, it's ultimately required to develop some kind of a contrast agents that can actually give you a different, the functional, the multifunctional aspects of a contrast agent. Wherein as we know, the nanotechnology has brought up several efforts or several properties of nanomaterials can actually give you different kinds of aspects, like different multifunctional aspects. One is we can say imaging. Yes, we can do with the different nanomaterials. For example, gold is being used as a CT contrast, and if you see iron oxide is being another FDA approved drug that can also be used for an imaging purpose, and it's given a predominant amount of results, and it also gave you a better diagnosis, disease diagnosis. And second one is the therapy side. Yes, we can use different nanomaterials, like say gold nanomaterials. It's actually used for photothermal therapy, wherein you induce a light and you try to produce some kind of a therapy. Yes, and the third one is, it's quite important, the antibody conjugation is quite important, which is not actually feasible in the conventional FDA contrast agents. So the antibody is the specific target of a tumor of the particular receptor over the cancer cell that's going to give you a better conjugation. So all these function, it can be conjugated in one single probe. Is that possible? And that's what we're going to see today. But as of now, we have just started with some kind of a feasibility study of our nanomaterials. We are trying to focus on the therapy and antibody conjugation in future. But as of now, we'll be looking at only the imaging aspects of my nanomaterials. Okay, in this we have taken two different elements, that is, one is the tantalum and another one is the europium. So what these two materials is going to do, what these two elements is going to do. So one is the tantalum, that is we have seen several research papers of or there have been a lot of research that's been happen in tantalum, which exactly tell you what is how the contrast agents can be developed for tantalum. Say for example, tantalum is an ultimate contrast for a CT imaging, wherein the tantalum has a very high X-ray attenuation factor of an atomic number, which has a very high atomic number similar to gold, wherein the gold is actually used as a CT contrast, and this tantalum and a combination of europium can be used as a contrast agent, wherein the europium is another material wherein we use it for magnetic resonance imaging, and also this europium is very famous for optical imaging. If you look at this image, this was published in 2011. O et al. Have been produced this tantalum oxide as an amorphous material. Yes, so they have developed a PEG polyethylene glycol Ritzy conjugated tantalum oxide, which going to eventually act as a CT contrast, and also it's going to act as a optical imaging. Yes, we can see the results of this particular publication, which came in 2011, and the another important aspect that we need to see is how these tantalum can be used as a CT contrast. And in case of an europium, we can see another example that is published in say 2014. Sudhindra has showed the dopant concentration of europium, the interaction of X-rays with europium, and he has shown some kind of a positive mechanism of producing a luminescence light when X-ray is excited on the europium material. So his results have shown that the europium can also be used as an optical imaging. So in my work, what we are trying to focus is on, we are trying to focus on three different aspects. One is the europium doped tantalum oxide nanomaterials, which can be used as a computer tomography, and these europium doped tantalum oxide nanoparticles can also be used as an X-ray excited optical imaging and also as an X-ray CT imaging modality. So one particle can be used for two different imaging modalities, one as for a X-ray CT and also for an fluorescence imaging. These are my results. So let me tell you how do I synthesize these tantalum oxide nanoparticles. So it's basically a simple microemulsion technique method wherein it has been reported previously. And this microemulsion technique, wherein we generate microemulsions, wherein you try to load these tantalum precursor into the microemulsion to form as a tantalum oxide materials. But before converting into a tantalum oxide nanoparticles, what we did is we just try to load some of the europium precursor that can eventually heat it to form, or you anneal it over a period of time, and then you get a different types of different luminescence of your nanomaterials, or we can say we try to get a combination of a CT as well as an optical imaging. But what happened is over the time the concentration of europium is going to increase, the problem in the fluorescence quenching is going to happen. So we are trying to focus on the different concentration of europium that can actually provide you different the contrast that is going to vary for europium material. Okay, So what we did is, once we added these europium and the tantalum oxide formation, so we are further heated for 100 degrees Celsius for 48 hours, which enables it to replacement, or it's going to be an addition of europium into the tantalum oxide nanoparticles. And the results of this tantalum oxide and the europium have been clearly shown that you can see two different spectra that can be obtained at, say, 250 nanometers, and also at 285 nanometers. The morphology of this europium tantalum oxide can be seen for both the control as well as the doped one. So wherein the control clearly shows you a spherical nature, but also it can be seen as an amorphous material, wherein the europium doped one, which has slightly a change in your particle size, but then the particle size is different based on the the dopant concentration. It's not uniform all over the tantalum oxide nanoparticles, but then you can see a small dark changes in your particle sizes, and also some kind of the dopant that is forming in your europium material. And this is your SAD patterns. We clearly shows that it's going to be an amorphous structure, and we have also studied the effect of the concentration of europium and tantalum oxide. Okay, so further the XPS clearly suggested that there is a presence of europium ions, and also the possibility of Ta2O5, the tantalum pentoxide, and also we have the TaOX nanoparticles, and we also have bondings of Ta and O. So all these results of XPS going to clearly suggest to you that there is a combinations of oxide materials of tantalum and also the presence of europium that's clearly going to tell you the europium is actually doped in the tantalum oxide nanoparticles. And another interesting thing about the europium doped nanoparticles is we need to look at the optical properties in order to understand how these efficacy of luminescence of these nanomaterials. Okay. First we look at the excitation spectrum, wherein you can clearly see the absorption of these europium compounds in the levels of, say, F2D, specifically if you are going to see there are 3 different absorption points, and also there is a 1, 2, 3, 4 and 5 different levels of the electron transition from LUMO to HOMO. So wherein we can clearly suggest that there is a enormous amount of the photoluminescence is being generated from the europium doped tantalum oxide nanoparticles. In other words, we have also seen the emission of these tantalum oxide, europium doped tantalum oxide nanoparticles in excited using an handheld UV lamp. That's clearly suggested that the red color luminescence, which is actually going to lie in the area of, say, 5DO to 7F2. So that's going to come under around 620 nanometers, wherein this absorption of 365 has been clearly seen at this range, that is 7FO to 5D4. And we can also see there is a difference in the electronic states that clearly suggests to you that the presence of europium is going to help you in the transmission of electrons from LUMO level to the HOMO level, wherein the emission spectra is going to happen. And finally, we have tested our nanoparticles in the presence of X-ray, when X-ray is going to exposed on a europium doped tantalum oxide nanoparticles, and that is going to be captured using a RGB camera, because the fluorescence that going to come from our europium doped tantalum oxide nanoparticles is going to be is not directly falling onto the RGB camera, wherein we have a 45-degree mirror that helps you in transmitting your light from the sample to the RGB camera. So this clearly we can try to avoid if there is going to be any kind of an RGB camera problem. And finally, we have seen with different concentrations of the different X-ray voltages of these nanoparticles has been exposed. So if you see this is an optical imaging wherein without any X-ray excitation, and next is 50 kilovolt and 100 kilovolt and 150 and 200. Surprisingly, we could see there is an increase in the enhancement of europium ions, the fluorescence that has been trapped by these the X-rays have been trapped by these europium ions, and it eventually converts into an red color luminescence, wherein the previous slide we have seen there is an emission spectra that is going to come from when you are going to excite at 365 nanometer UV lamp. Similarly, we have got around 600 nanometer specific color range when you are going to excite at X-rays at say 150 and 200 kilovolts. And here we have used a current of say 1000 microamps. This has been this image has been taken in a non-destructive testing system, which has a nanofocus and a microfocus tube, and we have used a microfocus tube at a current of say 1000 microamps with different kilovolts. So on a pay scale we can see on a scale we can see at a higher increasing the X-ray voltages is been increasing your luminescence. And I conclude, we have developed the europium doped tantalum oxide nanoparticles, and we could see we have characterized these oxide nanoparticles using a TIM, and we have understood the structural characterization of these nanomaterials, and also we have studied the optical properties, wherein we can clearly see there is an luminescence when you are going to excite at a UV region. And also we have seen the feasibility of the doped europium ions into tantalum oxide nanoparticles that clearly says that this particular contrast can be used as a Zeol contrast. We are trying to work on these nanomaterials wherein the tantalum is also enhancing this europium ions for a better luminescence. The work is actually under progress, and we are still working on how these europium is going to enhance the enhanced luminescence of europium is going to be boost up by the tantalum oxide nanoparticles. And finally I thank the institutes, Indian Institute of Technology in Madras and also SRM University for providing us an opportunity to present my work in BIGSS, that is Biophotonics and Imaging Graduate Summer School. And please be safe and stay home. Thank you.